Hello, everyone. I am Dipti Pitta. I work with the gut microbes that colonize the gastrointestinal tract of different animal species. I'm really interested in the mechanisms that underlie how microbes play an important role for improving productivity and reducing methane emissions in different ruminant species. In this particular talk, I'm going to talk how methane is formed, what is the role of microbes, how do we investigate microbes, and what are the strategies out there that are targeted towards reducing methane emissions in ruminant species. So, as we all know, ruminants have a unique ability to convert the indigestible plant material to usable substrates such as milk and meat. This is possible because of the presence of a complex multi-chambered stomach which allows for colonization of microbial ecosystem comprised of bacteria, protozoa, fungi, and methanogens that exist in a symbiotic balance with the host. To give a perspective of the bacterial numbers, there are about 10 to the power of 11 cells per mil of rumen fluid. So you could imagine millions and millions of microbes that allow for breakdown of feed particles in the rumen. So during microbial fermentation, these bacteria, protozoa, and fungi produce substrates such as short-chain fatty acids and microbial protein that is utilized by the host. In this process, they produce so large quantities of carbon dioxide and hydrogen that is utilized by methanogens to form methane. This methane builds over the period of time, gets accumulated in the gas pocket, and is released through eruption to the breath samples into the atmosphere, causing environmental concerns. It's not only an environmental problem, but nearly 10% of feed energy is lost in forming methane. So, either to improve productivity or to reduce methane in understanding of microbial population, what goes on in the rumen is important. So, because we want to reduce methane emissions from ruminant species, researchers have invested efforts towards developing new mitigation strategies that could be applied at the herd level as well as to individual cows. Some approaches that are targeting the herd are providing good quality pasture with very high digestibilities or providing good quality energy dense materials such as concentrates can reduce methane emissions. Of course, reducing cow numbers also is a great way to reduce methane emissions at the herd level. But when you come to individual cows, there are some supplements that could be added to cows' diet, such as lipids or secondary compounds such as tannins that can compete with methanogens for hydrogen. Also, there are supplements like ionophores that interfere with gram-positive bacteria and allow less methane formation. Some strategies are out there, like genetic selection, selecting for cows that have a natural ability to produce less methane or have the ability with high feed efficiency, all can reduce methane. Finally, not the last one, but the microbial interventions such as the use of vaccines to bind to specific methanogens or small molecules such as coenzyme inhibitors that can interfere with enzymes involved in methane formation. While these are promising, results have been inconsistent because we don't know what goes on in the rumen. Some of these microbes can adapt to these technologies, or sometimes in the process of increasing the dose of these inhibitors, there could be several adverse effects such as reduction in feed intakes or productivity or toxicity to ruminant species. So then what is the solution? We have the new technologies, the genomic technologies that could allow us to get deeper insights, deeper look into the rumen, where we can see now what sort of methanogens are present, how do they contribute to total methane formation at the gene level, who are the active methanogens, how do they contribute to total methane formation, And then when an inhibitor is provided, what sort of an effect does it have on different methanogens and other microbes? And most importantly, under methane inhibition, the accumulated products such as hydrogen or carbon dioxide, where do they go? Can energy be conserved within the rumen to help the cows to produce more milk? So these are some of the questions that we are trying to answer at my lab. So there are techniques measured to measure methane on dairy cows or ruminant species. The well-known technique is the use of animal chambers where animals are confined. So inflow and outflow air can be measured, can be collected and measured for methane. But this is not possible on all cows and requires sophisticated facilities. There are other techniques like tracer technique using sulfur hexafluoride as a tracer gas to measure methane. This requires again equipment, and there is so much of variability both on a single particular cow or between cows. And the most advanced technique is the green feed, the C-lock, that is invented recently and has the potential to measure individual methane measurements on cows. So this facility allows us to take the unit from one cow to the other, both indoors as well as outdoors, have the ability to capture the breath sample like for 5 minutes. So the cow has the head in the chamber, breath sample is collected, and so different composition of gases can be measured. Once the cow is outside of the chamber, the unit is left for collecting the external air samples before moving it to the other cow. As many as 8 observations can be recorded from cows in a single day to get an aggregate of methane emissions emitted by cows in a single day. Then what are the methods for rumen sampling? How do we access rumen contents? We have come up with a very cool technology to get rumen digester samples from the cow. We don't have to fistulate the cow. This method allows us to pass the tube using a speculum into the mouth of the cow. Against vacuum, we collect the rumen sample. As much as 500 milliliters of rumen fluid can be collected. This can take anywhere between 2 minutes to 6 minutes, which requires some restraint on the cow, but a good quantity of rumen sample can be collected. This rumen sample is filtered and we get the rumen digester sample on the right side. And this is the sample that we process in the lab to look for what sort of microbial populations are present and what is their role in methane formation. So using these cool technologies, we are now trying to investigate the effect of most promising mitigation strategies. The first one is the natural variation among dairy cows for methane yield. In collaboration with Penn State, we've screened more than 200 cows for their methane emissions, and we've identified a cohort of cows that are typed as high methane emitters and low methane emitters. Then we also have the opportunity to work with some of the potent inhibitors. One of them is 3-nitroxypropanol. This is a small molecule inhibitor that can bind to methyl coenzyme M reductase, an enzyme that catalyzes methane formation. And of course, we have the CVs, which is creating renewed interest among the community as well as producers. And this has the ability to knock down methanogens in the rumen. So the first step we did is we measured what is the difference in methane emissions per cows. So when we compared the high and low cows, we found that the low emitting cows can produce 22% less methane than the high producing cows. And when cows were given 3-nitroxypropanol, methane was inhibited by nearly 26% compared to cows that did not receive the supplement. And when seaweeds, Aspergopsis taxiformis is the variety that we used, when fed at 0.5% of the dry matter intake, we found that more than 60% of methane reduction in cows that received the supplement than cows that did not receive the supplement. So then we moved on to look at what's happening at the microbial gene level. And we picked the gene copies of the enzyme that catalyzes methane formation. And surprisingly, we found that the low producing cows, the genes involved in forming methane were reduced by 26% or were lowered by 26%. And then in cows that received 3-nitroxypropanol, these genes were inhibited by 26%. And when the product was taken off, they came back to normal. Similarly, when seaweeds was given, these genes were reduced by 60%, thus providing a molecular basis for what's happening at the cow level in terms of methane emissions. This is not the only thing that we are working on. We are also trying to come up with a new strategy to group cows based on microbial populations. We propose that certain groups of bacteria and archaea come together and that there is a natural variation in these microbial associations. So based on the microbial associations, we found three clusters. As you can see here, that in the red cluster, there is one sort of a microbial cohort that is present at a greater abundance than the one in the blue, which has a different set of microbial populations. So what we have found is that these microbial associations are linked to short chain fatty acids, thus indicating different mechanisms by which energy can be partitioned. For example, in the blue cluster, we found that the cows are more efficient and they can produce less methane. This procedure also allows us to reduce the inter-animal variation, and there are differences between different groups of cows. So by grouping these cows based on microbial associations allows us to group the cows based on efficiency. So we can select for cows that are more efficient based on microbial populations and reduce methane emissions. So not only this, we are trying to find novel indicators for ruminal efficiency by taking advantage of new technologies which are taking over humans as well as animal world. We are able to monitor or track the activity of cows. For example, the figure on the left, I show here a cow wearing a collar, which allows us to measure the rumination time, that is how many times the rumen actually moves and how many times it chews and how many times it stands and sits down. So much of information can be gained from this collar. For example, here, using the scholar, we get real-time data that can be captured based on day or between days or across several months. Now, using these measures, we can identify a health or a sick event or to measure how rumen responses are being done over the course of the time. For example, here we show that there are differences between cows based on this rumination time, and we can detect a health event. So using such sort of parameters, we have identified that cows can be grouped based on the rumination time, which is again linked to efficiency. So for example, the blue blue cluster here has a higher rumination time, that is the rumen can move faster, allowing for a different group of microbial populations to prevail. Similarly, the low rumination time are kind of encourages the development or proliferation of slow fermenting bacteria. So from these, we are able to say that rumen microbial populations rumination time are connected, providing a mechanism taking advantage of an advanced precision technology. So all these technologies are allowing us to understand the mechanism what goes on in the rumen, and from the study, cows that have a natural ability to produce less methane have a different microbial population and consequently different energy partitioning mechanisms in the rumen. Different supplements can be used as potent inhibitors. They have different mechanisms. So a molecular understanding helps us to complement different strategies to further reduce methane emissions from cows. And our results indicate the presence of different pathways by which we can bypass methane formation and help to conserve energy within the rumen. And we are taking advantage of different technologies, integrate those different types of data to help us to gain new insights on the biology of the rumen so that we can improve not only productivity but also reduce methane emissions from these cows. Thank you for listening.